So it's been a while. Where we left off was on Thursday. We started looking into, we're done with cells, and now we're looking into something called meiosis. So at this point, we've established that we know how cells split. They split through mitosis and cytokinesis and interphase. So we're looking at meiosis, which is different than mitosis. These are two different things, and they're very easy to mix up. So we've only just kind of scratched the surface with it, which is meiosis involves ensuring that things are done accurately, and it's another way to split our DNA. So again, we've looked at characteristics. We know that those come from genes, your hair color, your eye color, your height, medical conditions or lack of medical conditions come from DNA and the genes and your chromosomes. So there's 46 chromosomes, which have 23 pairs, all of which contain hundreds of genes. So there are tens of thousands of genes that make up you. So we first started looking out at homologous chromosomes, they are the same length, they have the same, this is called the centromere, and they carry the genes for the same trait. So both of these ones are maybe about your eye color, for example. So they're homologous if they're the same. So then we started looking at what's a haploid cell, what's a diploid cell, etc. But let's start getting into this. So we're going to start learning really how genetics get transferred up. So starting out here, we're going to start looking what the heck is meiosis? How is this different than mitosis? So we're going to look at how it's different, because if you're looking at this, it's kind of the same, right? This is why it's really easy to mix up the difference between this. So here's how it works. So again, mitosis is where it is, it's creating exact copies. So mitosis creates exact copies, as we discovered. Meiosis is a little bit different. So meiosis is a cell division that reduces the number of chromosomes in a cell and produce gametes. We have a couple different phases as well. So we're going to start out here with these different phases. So meiosis, the first phase is interphase. Just like before, we have interphase. It's going to have the same names. So in meiosis, this is where your chromosomes replicate. See your chat there. Thank you. So interphase is where the big thing here is the chromosomes replicate. So it's not much different at the start, right? Yeah, we're going to copy some DNA. This is where it's going to start to differ, though. And this is color-coded to help make it a little bit easier to distinguish. So what's going to happen now in prophase, and specifically prophase 1, is these chromosomes are going to start to pair. They're going to start to pair. Think about it again, for almost all living life, there's a man and a woman, they're each contributing their DNA. It's been coded here, blue and orange to kind of represent that. So prophase 1 is when these pairs start to form. After this is when it gets a little different. So here's where it starts to get a little different. So this is still under prophase 1. This is still the same thing, but so really pay attention to what's happening here with the colors. So basically what's happening here is these chromosomes are going to pair together, and then they're going to do something called synapsis, which basically means they're going to cross over. They're going to exchange some information. They are trading, is what's happening. All you really need to know is that synapsis is when these trade and they cross over. What does that mean? Well, when we have these segments, they're going to exchange some of these segments. Basically, they're going to exchange genetic information. Hey, I'll get this part, you get this part. You can see the difference here that happens. So we start to pair together. Right here is synapsis happening, right? So where I'm circling is where we have that crossing over, synapsis. And what's happening is they are exchanging the material. They have traded their material, and now we have entirely different DNA. We have more unique DNA. So it is not an exact copy anymore. There are slight changes to it. And this is what's going to make this is what's going to become the biological principle that's really all life is stemmed from. So again, so what we have here is we have two pairs of chromosomes, right? We have two different pairs of these, and they're starting to cross over, and it changes. It's now different DNA. They've exchanged parts of it. They've traded. So meiosis is really where we're generating new DNA. So now we follow through a pretty similar pattern, just like before. So just like the same with mitosis. So now what happens is just like before, those chromosomes attach to those spindle fibers and they line up at the equator. There's nothing different there between mitosis and meiosis. It's the same. Okay, so far throughout this entire process, so meiosis versus mitosis. Again, meiosis has been the exact same with the exception of prophase 1, because we have that crossover. Metaphase is the same, interphase is the same, anaphase is the same. So there's a lot written on here, but if you remember what mitosis is, you don't really need to know much about this, right? These chromosomes split and they get moved towards the poles of the cell, and then eventually the cell itself is going to split, right? Again, there's only one phase here that's different. Otherwise, it is the exact same. So mitosis has the exact same DNA, and meiosis has those differences. You're looking at that, you can see that it is different genes being passed along. And if you're looking at it, we've now gone from 2 blue ones, two orange ones, to now we have 4 different ones. These are all different locations on here. Does anyone here have like cats that have had kittens before?
Yeah.
The cats don't always look the same, right? Sometimes like the kittens have different fur coats and stuff. Sometimes they might not even look like their parents, or they're like a mix. This is how that happens, because this genetic information gets mixed up a little bit. Again, they trade. So just like before, telophase, it starts to separate, cytokinesis occurs, etc. And that's the end of meiosis 1. So you don't need to take some super detailed notes about this. You just need to know that prophase is when we're starting to get ready to, the DNA starts to get ready to copy itself again. And so again, if you remember our acronym, popsicles melt like at temperatures. We're at prophase. What's the M stand for again? Test our memory. Metaphase. So next is our metaphase part. And again, it is just like before. Metaphase 2 is just like metaphase 1. It's just like mitosis is metaphase. The chromosomes line up and they get ready to split, right? They line up, these apparatuses attached to them. Nothing's different there. So you don't need to do a full repeat of your notes. The one thing that is important to know is that meiosis 2 has a haploid number of chromosomes, which we'll get into. So again, metaphase is identical. The only difference is there's 2 cells here, but as long as you remember the principle of it where it splits up, and in meiosis 2 it involves 2 cells. So next we go into anaphase, and again, it is the exact same thing. It's the exact same. I'm not even like, you guys don't have to worry about writing this stuff down here. Just know anaphase 2 exists. It's done the exact same way. They start to get pulled towards each pole of the cell. It's just happening to 2 cells instead of 1. Okay, so what actually happens here, I know I'm going to end up in 4 cells. I have 4 pairs here. So these are going to eventually become paired together, which are in our last phase here, which is telophase. So these reach the poles, and again, the nucleus starts to kind of form. We're going to split into 4 cells now. And again, you can see how now what I have is I have 4 different unique cells. That's all different DNA, right? If you're looking at the color of them, we have entirely different things. Now, these are still the original ones, but they are unique still. We have genetic diversity. So again, the telophase 2 is the exact same thing as telophase 1. And finally, what's going to happen is we're going to go through cytokinesis. This is when it actually splits, right? So now they are themselves 4 cells. They haven't split here. They're about to. Now they've split. And so what's happened is we've created 4 haploid cells, and they each have their chromosomes. So what we're left here is we're left with 4 haploid cells, right? Haploid cells, as you can see here, they just have there's two halves of a chromosome. A diploid cell, though, has it has full chromosomes. So I've gone from 4 complete chromosomes to 4. It's still 4 complete chromosomes, but it's split into 4 different cells. So we split into 4 different cells. The other question I have for you is, what's the difference between mitosis, which is something we learned last week, and meiosis? They have the same phases, so why are they different? So the big one here, so mitosis, the first one, it just happens once, right? Mitosis is just it. Mitosis is where we have one cell and it's getting ready to turn into 2 cells. Kind of looks like that, right? Meiosis, however, has both, there's 2 divisions. There's meiosis 1, meiosis 2, and also it's creating cells that are not genetically identical. So the difference is it creating an exact copy, or is it creating something that's close but new? So the big thing here is that meiosis is what results in genetic variation. This is why you can take 2 dogs, you can breed them, and you'll get a hybrid between the 2 of them. The big difference between this is mitosis creates identical cells, meiosis creates different cells. And this is how we get how genes pass on. With one other step, but that'll come next week. Let's see what we all need here. I don't think we're gonna need much of that. That's all gonna come next week. We're gonna skip this. This is something that's gonna come, but these here, don't write any of this down. But these here are genes. We're gonna learn how genes pass on and the combinations that can come from it. This is just talking about, hey, meiosis provides variation. That's all we need to know right now. We'll learn specifically how we can predict what happens with it probably next week. One thing that's just important to note is that not all creatures do produce sexually. Some creatures produce asexually, aka it gets everything from its single parent. If that's the case, it's identical. But if you have sexual reproduction, you have 2 or more organisms contributing DNA, you change the animal, right? You have introduced new DNA, you've created a hybrid. Okay, so we've learned a lot about this stuff. There's a whole lot. We're very shortly going to be learning. Oh, perfect. Thank you. We'll very shortly be learning specific genetic stuff and how that gets passed on. But I really just quickly wanted to go over what some of this stuff was here again. So I'll give you, you don't need a lot. I don't think you need all of it. Don't worry about question 2. Scratch question 2. You only need like a line for each one. Line or 2. Not a lot. So let's put a nice little bow on this. We're just going to go through the 3 questions. So how does meiosis produce a haploid gamete? Reductive division. It reduces it and it divides it, basically. Right? We go from We go from 2 to 1. So our next part here is, no, I'm gonna put that. Synapsis. What the heck is that? How does that occur? Basically, those chromosomes, homologous means the same. If I give you two blue and two pink, they touch each other for a little bit. I now am going to have genetically different things. They trade. Synapsis is the trading part of it. You don't need to go much deeper than that. They touch each other, they exchange material, and they unbind. Okay, and then our final one here is, how does meiosis create genetic variation? Well, mitosis does not. Basically, meiosis creates a new combination from the parent, but mitosis is a clone. So it's cloning versus a combination.